What's up, guys? Running crazy, man. Okay, I'm fixing to run through quickly chapter 11, try to get that down for us. I'm going to share my screen and we'll walk through these PowerPoints. This is one of my favorites. Hopefully we'll get to lecture a little bit of it today. Okay, you should be seeing PowerPoints here in chapter 11. Let's see. Okay, all right, here we go. Now, first of all, here's what we're looking at. We're talking about Gregor Mendel today, who's one of my favorite scientists of all time. So we'll talk about him and his laws, and then some ideas beyond his laws. Okay. A little icebreaker. When at the beginning of the chapter, they talk about phenylalanine, which is one of the amino acids, and it's one of the two amino acids that's in NutraSweet. And so this is a disease, phenylketonuria, which can cause they're missing an enzyme to break down phenylalanine, and excess accumulation of this causes nervous system disorders. And these children that get this disease have very dangerous mental problems where they cannot function normally and don't actually, I believe it is, their mind doesn't develop much past the age of 10 or 12 years old. But that's why the phenylketonuria label is on all NutraSweet food. Some interesting side notes are on that one. Okay. All right. So who is Gregor Mendel? First of all, let's just say this is one of my favorite people in science because he is considered the father of genetics, and he's considered the father of genetics, and he didn't. He died unknown as an unknown monk. No one ever knew he did anything great, but he published his results, and then later on, after his death, they were discovered, and post-death, he is considered the father of genetics and one of the key people, one of the big scientists of all time, I guess, in the history of science because of this. Okay, so what he was looking at was at the time of his research, when he was doing this research in the 1860s, the concept of blending was kind of the norm. It said that basically that blending was how genetics works, hereditary works. Okay, and if that was true, it would actually, everybody would be the same over time. So it's kind of now that we now think about that, but we didn't know that. It was a very popular time, and Mendel wanted to do some experiments to try to disprove this concept that everything was blending. That means if ugly people marry ugly people, they have ugly kids, right? Two tall people are going to have tall kids. Tall person, short person have an intermediate kid. So everything should lead to intermediates, okay? All right, let's take a look at what he did. He was an Australian monk. He did study mathematics, science and mathematics at the University of Vienna, okay? And some other experiments have been done before. So he was garden pea was a good experimental test organism. Nobody cares if you make them do things they don't want to do because they don't have a mind, right? And he formulated these laws of heredity in the early 1860s without any knowledge of the cell or chromosomes. He did not have a microscope. He had a lens to look at things, but not a microscope. And these experiments on inheritance of simple traits, the garden pea are going to disprove this concept of blending. Okay. So here's what he did. First of all, the garden pea is a good choice. Okay, easily to be bred. It has both male and female parts on it. Has both anthers and stamens and female stigmas, right? And so they have both sexes. They have dual sex on each flower. And he would use his little lens and his little tweezers to castrate, I guess, to remove the male genitalia to force breed the plant with who he wanted to breed with by putting the anther into the stigma of another plant. And in doing this, he was able to look at several traits at one time. Now, in his terminology, the word gene wasn't known at the time. So instead, he used the word trait. Okay. And when he says something is true breeding, he means that it is, if you got blue, you always, or green in the case of these, you breed them together and they're always green. That really what we now know would be an homozygous, means everybody's the same. And if someone's different, they're said to be heterozygous, if they have two different copies of a gene or trait. Okay, his first experiment, he was going to use P stands for parental generation. F1 is for the first generation of offsprings, and F2 is for the second one. And so what he did is he developed multiple gardens of what he called true breeding plants. And what we now know is that they were all homozygous dominant for that trait. In other words, if he had tall plants, he put them in tall and bred them together. And if he were to get a short, he put them in the short garden like this one. So he had these true breeding short plants. Now, if blending is true, then you should get an intermediate. And when he bred these together, what he got was, what we now know is that they had two dominant genes, which we do use capital letter T, okay, for tall, and little t would be short. And so basically all the sperm or egg that they could pass would be the big T here, and that made all their children what? Not intermediate, but they were all tall. And then all of these, the breeding of these true breeding talls and true breeding shorts led to everybody being tall. And he came up with this concept. This is where genius kicks in, that there were two traits in there were two competing versions of every trait or gene, as we'd call it now, one from mom, one from dad, and those two compete for what's going to be expressed in our phenotype. So phenotype is your physical expression. Genotype is the genes you have. So this one, sperm, this one had big T genes, and this one had little t, and both of their gametes or sperm here or egg were big T's and little t's from this group, and they come together and form this heterozygote. Now to double make this super exciting, then here he's doing it again, showing it here. We can do a Punnett square. Now later on, a guy named Punnett came up with this square that you could use instead of doing just by probability. But you can do this by probability because 50% of the time you're going to get the big T here, 50% of the time you get the big T here, which means all the time getting big T. So here you can see that in this generation, he's going to breed these together. Now he's got all these that are what? Tall, F1s. And so his next experiment is to breed them, little incest here between brothers and sisters here. But he breeds them to each other. And each of them would have big T 50% of the time, little t 50% of the time. And the other, the female, or that would be the female here, and the male would be the same. Then you put them in here. You just stick them in together. So big T here, big T here, big T here, little t here, big T here, little t here, little t, little t. And what you end up with is 4 genotypes. Big T, and one out of 4, 25% of the time. Big T, little t, 50% of the time, 2 out of 4. And little t. So when you do this, you actually get the recessive trait 25% of the time. And when he did this experiment, he's looking for a 3 dominant to 1 recessive. And by the way, check out his numbers here. Here's his numbers, man. Did he knock out a home run here? For stem length, he got a 2.84 to 1 ratio. For pod inflated versus constricted, which is recessive, he got 2.95 to 1. Seed shape, look at that, 2.96 to 1. Seed color, with yellow being dominant and round, 3.01. Flower position at the axil or terminal, 3.14. Flower color, purple to white, 3.15. Pod color, 2.82. So his overall, look at his N number. The number of individual traits looked at was 20,000, and he got a 2.98 to 1 ratio. Absolutely grand slam home run, looking at 1, 2, 3, 4, 5, 6, 7 different traits in the garden peas. Okay, so from that we get his classic law of segregation. Each individual has a pair of factors, which we now call alleles for each trait, which we now call gene. The factors, alleles, segregate separately during gamete, sperm, and egg formation. Each gamete contains only one factor and one allele from each of the pair that they got in them, one from their mother and one from their dad. Fertilization gives the offsprings 2 factors for each trait, resulting that was the result of his monohybrid cross. And that's one of the great discoveries of the history of science, this experiment, because this is the fundamental key to hereditary. We have now on our 46 chromosomes like 23,000 genes, 2 versions of each gene, one from mom, one from dad, and those 2 compete in a dominant recessive type trait. Fascinating. So he's primitive with that. Okay. So therefore, each trait in a plant pod is controlled by 2 alleles. Dominant masks the expression of recessive. Alleles occur on a homologous pair, right, at the same locus as where it's at. If you have 2 identical genes, like big T, you're homozygous. If you're big T, little t, then you would be heterozygote, meaning you have different alleles. Okay, and here's those alleles on chromosomes, right, and they're copied during interphase, so we get those duplicated chromosomes coming into meiosis. Genotype is the 2 genes you have. Homozygous genotype would be big T, or little t, or whichever letter you want to use. And phenotype is your physical expression. I just said that. Okay, the dominant recessive allele represents DNA sequences that code for protein. Dominant alleles code for the protein associated with the normal gene function in the cell. Recessive allele represents a loss of function of that one. Okay. During meiosis, no one's going to separate and the 2 alleles divide. Remember, we talked about that. His second law, then he did a dihybrid cross, looked at 2 traits at once. Okay, so he's going to look at 2 traits at one time, and he is going to track them. And from this, he's going to come up with his law of independent sort, which we now know is not always true because of crossing over and all. But look at that: the pair factors for one trait separate independently of the factors of others. All possible combinations will occur. The F generation is the parental generation of breeding experiment. The F1, the same little things we talked about. So here we're going to look at 2 traits, and he used 2. He had to garden with 2 dominant traits, of tall and green versus dwarf, little t, and yellow pod. Okay, little g. Now that means that when you look at that, then everybody in this generation, all their sperm are going to be big T, big G, and all their sperm or egg is going to be big T, little g, excuse me. And so if you breed them together, everybody's going to look just like the dominant parent, but they're double heterozygotes. Big T, big G, little g. And then he did a Punnett square. He actually did it by probability, but then later on we can draw it more visual in a Punnett square. And look at this Punnett square. So you end up with 16 different potential children, right? By doing 4x4 grids here. And you just add them in here like big T, little big G here. And look what happens. You end up with 9 individuals that look like the dominant parent or like their parents. 3 that have the dominant trait and the recessive trait, and then 3 that flip it the other way. So these have the tall and yellow, and then those 3 out there in that shade, and then these 3 here have the dwarf with the dominant green, but 1 out of 16 have both recessive traits of this grandparent. Pretty wild when you think about that. Now, I don't want to make a huge speculation here because we have how many traits? Not 2, but 23,000 genes. But I think what this tells us is that just real big, extra, probably not very scientific, but just a way look at this is that if you are, I'm just saying, look at this. It's just kind of fun here, but just think about this. So if you're normal, okay, and for 2 traits, if you're heterozygote for 2 traits, and your husband or wife is heterozygote for 2 traits, then the kids, and I'll give you 2 of ours that all work like this. Being able to curl your tongue, you can make that big T, and the earlobe, big earlobes would be dominant too. That'd be like big G and big. I'd be big T, big G. And so if you and your wife were heterozygote for both, you would have 9 kids that could curl their tongue and big ears, 3 with big ears and can't curl their tongue, 3 that can curl their tongue with straight ears, and only 1 out of 16 look nothing like the parents. Something to be said about that. I don't know how much I'd extrapolate into that, but I find that pretty intriguing. And here it's just going to show how that independent assortment occurs, right? And all the different combinations you can get. And those are how many you can get. Look at the different combinations you get when looking at 2 of those traits. There's your different sperm and egg there. Pretty cool. Showing how independent assortment works. Another one showing that from the chapter on meiosis. Because we already talked about Punnett square. You can do it by probability. You flip coins and figure this out too. Because the chance of each of that is 50%. Okay, so you can do it either way. Here we'll do another one here, or as a trait that's dominant, normal pigmentation versus lack of pigmentation. Both of these individuals are carriers, and they have 3 children with normal pigmentation and 1 that is an albino. You can see how that would work. Punnett squares can be very helpful. Mendel also come up with a test cross where you breed somebody back to a homozygous recessive individual, okay? And that will let you know if what that person is. Because we're not sure if this person right here is homozygous dominant or heterozygote. These 3 we're not sure about. So you breed them back to a homozygous recessive, little a individual, which we wouldn't do that in humans. That would be unethical, but you could do it in animals or in plants. And look what happens. So he did this. So these guys, he wasn't sure, right, what this progeny was all big T, right? But he thought, right? So what he did then was he bred them to the homozygous recessive trait. And this one, if you breed that one, then you always get what? Everybody will be heterozygote, okay? Get that? So if you breed an individual, this individual to the homozygous recessive person, you're going to get that. If you breed them to a heterozygote, you would get a different ratio, right? So you would get, if they're a heterozygote, you'd get half tall, half short. You get half and half. There's what it is. So A is breeding them to that. Sorry. This individual bred to our homozygous recessive will give you half tall, half dwarf. Here, everybody's tall. Okay, that's pretty cool. So test cross is breeding somebody back to a homozygous recessive, and we can see they're either that, and that tells you that half are tall, half are dwarf. That means that person was heterozygote. If they're all tall, that person was homozygous dominant. Sorry about that. We're not seeing that screen real good. Okay, and then he looks like, then we look at some things, some human diseases. Autosomes are the first 22 pairs, and the 23rd pair is the sex chromosomes. Okay, so some genetic disorders can be autosomal dominant, means they're on one of the first 22 pairs. And look at that, individual with double A will have the disorder, and individual with big A, little a would have the disorder, and only a little a will not have the disease. Recessive disorders, you'd have to have big A don't have it, big A, little a don't have it. You'd have to be little a to get the disease. Okay, here's a couple of diseases of this. I believe this, and they don't label these. This showing you just how they would look in a pedigree. So an autosomal recessive disorder right? Everybody would be a carrier. Now, if that person was heterozygote, then half would get it too. So that's pretty, this is actually how, so a lot of times it jumps generation and pops up from generation to generation. Heterozygotes are unaffected. These are some good traits about each of those. Close relatives who reproduce more likely to have affected children. Both male and females have equal frequency. That's pretty cool. Although dominant, if you get the gene, it keeps fasting straight down. Two affected parents can produce an unaffected child if they're both heterozygotes. And we saw that right here. But these children, half the children, well, 4 out of 5 were fine, and then 2 had the disease. So we look at some of these patterns. Some autosomal recessive disease include methylmetaglobulin, which we'll see a picture of that in just a minute. And cystic fibrosis here is the pigmentation on the right. It's not right. That's that one. Let's have that go to voicemail. Sorry about that. And then cystic fibrosis is the terrible where we have an error in making chlorine channels. And this is these kids choked to death, right? From their mucus buildup in their lungs. Pretty bad. Other ones here, here's some other ones. Osteogenesis imperfecta is autosomal dominant. I have a good friend of mine that had this. He did die at 53. And another one here, not familiar with. And then beyond, I mean, let's look at this. You can have multiple alleles. ABO blood typing, there's an A and a B gene, and then you can have 2 O's. So if the 3 different genotypes, or 3 different alleles, ABO, excuse me. Okay, that's pretty cool. So we call the ABO blood typing codominance because A and B and O are all codominant. So A people have to have both, have an AA or AI, BB or BI, and AB, or have to have 1A and 1B, and that's the rare blood type. And then O is just its own gene that have no A or B, it's a blank, it's OO genes. Okay, and O is actually more common, it's more common out there. Incomplete dominance is another one that we see also in nature where you get a blending. So this would be the blending one, and the classic example of is the pink roses. If you breed 2 pink roses together, you end up with 2 pinks, 1 red, and 1 white. Okay, with R2 being the white gene. So that shows you incomplete dominance. Okay, others, examples of this is familial hypercholesterolemia in humans, which was discovered in the late 60s. And this one where you have children that have no genes have totally no cholesterol receptors, and they have a heart attack usually before the age of 20. Those who have 1 gene and 1 normal gene have cholesterol levels of like 800 and then have a heart attack in their 40s and 50s. And then normal would be to have that heart attack in your 60s, 70s with cholesterol around 180. Another concept is incomplete penetrance. And what that means is some dominant genes everybody don't get it, or you may get a different degree of it. And a good example of this is polydactylism, where you may have the gene and not have any extra fingers, but then somebody else may have 5 or 6 extra fingers. So it's not, and that can give you levels of penetrance. Okay. Another weird concept here is this one called philotrophy. Occurs when a single mutant gene affects 2 or more distinctive, seemingly unrelated traits. Classic example of this is Marfan syndrome, which is a connective tissue problem, right? And look what it causes. It's called skeleton problems, causes aneurysm. That's what usually kills on the heart. Eye lens dislocate, severe nearsightedness, heart, blood vessel problems, skeletal problems, lung collapse, and skin stretch marks and skin stretching of the membrane holes around the spinal cord. So all this from 1 gene, it's a connective tissue gene. So that is philotrophy, when 1 mutation gene can affect more than 1 physical trait. That's an example of that one. Beyond Mendelian genetics, and this is something really important to bring out at this point, is that most of our 23,000 genes are more than 1 gene, 1 location. So we only have just a handful in the humans that's 1 gene. And those include tongue curling, earlobe, and Willis feet, just to name there's about maybe 10 or 15 you can name. Okay, things that are complicated like skin color, height, eye color. I won't even talk about eye color, it's so crazy, but intelligence, personality, think about IQ. These are multiple genes, maybe 10 or 12 determines intelligence, okay? Now, one of the coolest things about this polygenic inheritance that I think is really cool is if you go ahead and measure it, you will get a bell-shaped curve of the mathematical distribution of that. So in this particular one, we've got something that is 6 genes, and you can look at that. And so these people in the middle are normal, like height. And then you have very few short people and very few tall people. So you'll get a bell-shaped curve. And there are people in biology science that are so in love with this continuous variation polygenic inheritance that they will set their grades that way. So I could take any set of grades and put them in a formula and make a bell-shaped curve out of them, where you'd have what? Mainly C's, B's, D's, and very few F's and very few A's. Actually took organic chemistry with a teacher that did this, and it was very weird because all you had to do was just make right above class average to make a B. But it was almost impossible to make a. You make a 90 and get 2 points. Make a 50 and get 20 and go to 70. So I'm not a big fan of using bell-shaped because I don't want to determine every class is different. You class of 20 or 30, you could have an outlier that's all geniuses, or you can have an outlier that they're all Forrest Gumps, and they're just not going to do well. Okay. Also, we got X-linked inheritance. Okay. In this, there is a difference because women have 2 X's and men have 1. And so if there's an X-linked disorder, then it's more likely to occur in males. Okay. So they can look at this. So we use this, show some examples of this in fruit flies. And here's one possible showing extension of the range here in skin color if there were 3 genes for it. And you would have albinoism here and extremely dark skin and everywhere in between. So that's another showing that continuous variation. The X-linked inheritance is just looking at some on the X chromosomes. And so if it is on the X chromosome, then the male is more likely to get it because he only has 1 Y. Okay. So here's some characteristics of this. Some examples are color blindness, muscular dystrophy, okay, adrenoleukodystrophy, another wild one, and hemophiliac. I think most people have heard of hemophiliac running through the royal family, and color blindness. Here is I believe we got Queen Victoria's family. Look at her family tree. She was a carrier, and look at all those guys that had hemophiliac over 1, 2, 3, 4, 5, 6 generations, all the way down to the grandkids today. There's William, Kate, and Harry, et cetera, et cetera. Okay, pretty wild. Charles there, number 11 there, right? So that's pretty cool. So they have hemophiliac because they kept marrying first cousins. Look at that. Look at all that inbreeding going down through there. No wonder they messed up. Oops, did I say that? Sorry. So X-linked recessives are males. More males than females are affected. Affected sons can have parents who have the normal phenotype. For a female to have the characteristic, her father must also have it. Mother is a carrier. Characteristics also skip a generation from grandfather to grandson. If a woman has a characteristic, all of her sons will have it. So here is a grandfather. He gives it to his daughter, who is the carrier, right? And then her son gets it, and he actually married someone that was normal, okay? And here we can see it jumping through here. So all the daughters are, the daughter here is a carrier and her grandson. So jumping from father, his daughter's fine, and it goes, or his daughter or son has it here. In this case, what happened there? He had a normal son, but the daughter was colorblind, had the disease. So got a while there. So he usually jumps in there, it is jumping that generation down through there. Pretty wild. All right, that's all I got on chapter 11. Y'all have a good one, and we will try to get catch back up. I'm going to try to do some 12 here in just a few minutes. Thanks. Y'all have a great day.